Hello everyone, this is Dr. Vaishnavi Kanji. Today I am going to teach you practical of estimation of hemoglobin. Okay, so let's start it. First I will teach you some theory aspects and then I will show you how to do practical of estimation of hemoglobin. So now tell me what is hemoglobin and what are its components. Hemoglobin it is an conjugated red protein which are found in RBCs, red blood cells, and are responsible for transporting oxygen in the vertebrates, right? And its molecule it comprises of four polypeptide chains which containing two alpha and two beta chains. Now here types of hemoglobin. There are three forms of hemoglobin. First, adult hemoglobin. Second, fetal hemoglobin, and third, embryonic hemoglobin. Adult hemoglobin it consists of two, AHBA and HbA2. HbA it contains what composition? Alpha 2 and beta 2. HbA2 it contains alpha 2 and delta 2. Okay. Now fetal hemoglobin. It has two parts, HbF and HB bars. HbF it contains alpha 2 and gamma 2. HB bars it contains gamma 4. Okay. Now embryonic hemoglobin it has three types, HB grower 1, HB grower 2 and HB Portland. HB grower 1 it consists of epsilon 2 and zeta 2. HB grower 2 it consists of alpha 2 and epsilon 2 and HB Portland it consists of zeta 2 and gamma 2. Okay. In previous slide, we have learned about types of hemoglobin and now we will see the difference between adult hemoglobin and fetal hemoglobin. First point, adult hemoglobin, it contains of four polypeptide chains, that is, you already know 2 alpha and 2 beta. And in fetal hemoglobin, these 2 beta chains, they are replaced by gamma chains. So, it has 2 alpha and 2 gamma chains. Second point, it tells that adult hemoglobin at the time of birth only 20% is present and fetal hemoglobin 80% is present at the time of birth. Okay, and adult hemoglobin it appears in red cells of the fetus in 5th month, and this fetal hemoglobin it disappears by 5th month after birth. Lifespan of adult hemoglobin is of 120 days and that of fetal hemoglobin is only about 2 weeks. Adult hemoglobin it has usual affinity for oxygen and fetal hemoglobin it has higher affinity for oxygen as it binds to 2,3 biphosphoglycerate less avidly. And now percentage saturation at partial pressure of oxygen of 20 mmHg. In adult hemoglobin it's 30 to 35 percentage and in fetal hemoglobin it's only 70 percentage. What are the normal values of hemoglobin in adult male, adult female, and in newborns? Two things you have to keep in mind, that whenever someone asks you that the normal values, you have to tell in the range, okay? And don't forget to tell or write this unit, that is gram per deciliter of blood. Okay, so what is the normal value of hemoglobin in adult males? It's 14 to 18 gram per deciliter of blood. In adult female, 12 to 16 gram per deciliter of blood, and in newborn it's 16 to 22 gram per deciliter of blood. Okay. Now we will learn about few hemoglobin complexes. First, what is oxyhemoglobin? It is a bright red substance which is formed by combination of hemoglobin and oxygen. Okay, and it is present in oxygenated blood. Second, carbaminohemoglobin. It is a compound of hemoglobin plus carbon dioxide, and it is one of the form in which carbon dioxide exists in the blood. Okay. Third, carboxyhemoglobin. It is the stable complex of carbon monoxide and hemoglobin that forms in red blood cells upon contact with carbon monoxide. You have to see here, it is carbaminohemoglobin and this is carboxyhemoglobin. Okay. Fourth, methemoglobin. It is an abnormal hemoglobin in which iron is oxidized from its ferrous state to ferric state, that is Fe2 plus to Fe3 plus state. Okay. Fifth, sulfhemoglobin. It is an abnormal hemoglobin complex formed by the action of some drugs and the chemicals such as sulfonamides. Okay. And last, sixth, cyanide hemoglobin. This is formed by the action of some chemical called cyanide. What is an hemoglobinopathy? They are the group of disorders which are passed down through families, means they are inherited, okay, in which there is an abnormal production of hemoglobin molecule or abnormal structure of hemoglobin molecule, okay, like example sickle cell hemoglobin, thalassemia, okay. Now here comes the practical part. What is the most common method which is used for estimation of hemoglobin? That is Sahalis acid hematine method. Okay. Now what is its principle? It is the amount of hemoglobin that can be estimated by conversion of known volume of blood into acid hematine by addition of dilute HCl, okay, and subsequent color matching with a suitable given standard. What are the apparatus needed to do this practical? First, Sahalis hemoglobinometer, which comprises of comparator fixed with two solid standard colored glass rods. Second, hemoglobinometer square tube, then hemoglobin pipette and stirrer, okay? And second, N/10 HCl. Third, distilled water, and fourth is the dropper. Here are the images. First one is hemoglobinometer tube, which is present in the left side. Then middle one, it is an hemoglobinometer pipette. And the right side, it is an comparator box with two solid standard colored glass tubes. These are the images which are taken from our practical lab. This is an hemoglobinometer tube with stirrer. Can you see that? And here is the hemoglobinometer pipette, left side, and right side it is an comparator box. Okay. So here is the procedure. I will teach you first the procedure and then I will show you how to do the practical. Okay. So first take N/10 HCl in hemoglobinometer tube with the help of dropper up to its lowest mark, okay, and then hold the mouthpiece of pipette between lips. Prick the finger under aseptic precautions. Hold the pipette horizontally. Apply its tip to the drop and draw blood in it. Take care that there should not be any air bubble present in the pipette. Now transfer blood from pipette into hemoglobinometer tube in which N/10 HCl is present. Okay. Then rinse the pipette properly and avoid foaming. Now mix the content with the stirrer and leave it for 10 minutes for conversion of hemoglobin in blood to acid hematine. Okay. Now after 10 minutes dilute acid hematine by adding distilled water drop by drop and keep mixing it with the stirrer. Okay. Continue this dilution till its color matches with standard box. Okay, standard color, and then note the reading. So here is the practical video. First we have to prick the finger under aseptic precautions, that is we have to sterile the finger. Okay, now we have to prick the finger, ring finger. Then we have to wipe out the first drop. Okay, then make this second drop properly. Blood drop should be present properly, okay, so that it would be easy to suck inside the pipette. Now this is an hemoglobin pipette. Okay, we have to draw the blood till that mark. Now keep this inside the hemoglobinometer tube and drop inside that blood inside this tube. Okay, now mix this with the stirrer, mix this solution with the stirrer. and keep like this in the comparator box for 10 minutes. Okay. Now after 10 minutes we have to add distilled water drop by drop as it is shown in the left side of the pic. We have to add distilled water drop by drop in the acid hematine solution. Okay, and we have to add dilute the solution till that it matches to the standard comparator box, standard comparator color. Okay, like it is shown in the right side, and after that note the reading. Are the advantages of Sahali's method? First one, it is very simple, quick and easy, as you have seen, okay? And it doesn't require any costly instrument, and its cost is very minimal, so we can also use it for mass survey. What are its disadvantages? Like it is turbidity, second carboxyhemoglobin and methemoglobin, they cannot be estimated by this method, and the color of this comparator box, they may be faded. Precautions to be taken while doing the practical. Okay, first one, do not squeeze the finger to take out blood, otherwise tissue fluid might also come out. Okay, second, distilled water should be added drop by drop to dilute acid hematine, otherwise over dilution may occur. Third, the matching of the color should be done only against natural source of light. Okay. Fourth one, blood from the pipette should be transferred quickly into the tube containing N/10 HCl to prevent clotting of blood in the pipette. Now we will see few other methods through which this estimation of hemoglobin can also be done. Okay, first one is Tolquist method. It involves direct visual matching of the red color of drop of the blood on filter paper with color standard on the paper. Okay. Second one, Hendon's method. In this method, hemolysis of red cell is produced by mixing blood with a hypotonic solution like distilled water. Third one is sign methemoglobin method. The principle of this method, it lies in the conversion of hemoglobin to cyan methemoglobin by addition of potassium cyanide and ferric cyanide. Fourth one, Wu's alkaline hematine method. This method is based on the conversion of all hemoglobin and hemoglobin species into a stable end product by an alkaline solution of a non-ionic detergent. So this is the end of practical, and now these are the few questions that you should know at the end of this module. Okay, first one, what is the principle of Sahali's acid hematine method? Second, what are the types of hemoglobin? Third, what are the direct and indirect methods of hemoglobin estimation? Fourth, what are the advantages and disadvantages of Sahali's acid hematine method? Fifth, what are the precautions that we should take while performing this practical?